What if I told you CRISPR Therapeutics may have just built something that leapfrogs the entire gene editing field? A system that can correct DNA inside your body with pinpoint precision without bystander edits and at doses so small it almost sounds unreal. That's what the company claims with its brand new SyNTase platform, and its first therapy, CTX460, could probably set up a direct showdown with Beam Therapeutics in the race to cure Alpha-1 Antitrypsin Deficiency or AATD. Hey all, welcome to ShareTech, where science meets investments, and I'm your host Raj. Friends, today we are looking at one of the most exciting developments from CRISPR Therapeutics in 2025. Their SyNTase platform and first program CTX460. This technology could redefine what's possible in in-vivo gene editing, but it also enters a fiercely competitive market where Beam Therapeutics has already got human data. But before we dive in, if you find this breakdown helpful, please hit the like button and subscribe if you haven't done so. It helps the channel as well as it helps other investors like yourself discover this channel. That said, let us talk about the new SyNTase platform. So what exactly is SyNTase? Well, CRISPR Therapeutics calls it a next generation gene editing platform designed to go beyond current prime editing systems. Here is what makes it special. It uses a compact Cas9 nickase paired with a synthetic polymerase that can read chemically modified templates. Instead of long and fragile PEG RNAs, SyNTase uses shorter tag RNAs that are more stable and easier to manufacture. The system mimics natural reverse transcription, allowing it to make any nucleotide correction precisely without off-target or bystander edits. That's important because traditional gene editors often struggle with accuracy and unintended changes. SyNTase appears to solve that problem. Unlike the ex vivo cell therapies that CRISPR Therapeutics has worked on before, SyNTase is designed for in vivo use, meaning the editing happens directly inside the body. It's a one-time treatment delivered through a simple IV injection using lipid nanoparticles or LNPs, the same kind of delivery system used in mRNA vaccines. This approach bypasses the need to extract and reinsert patient cells, reducing complexity and cost. CRISPR's preclinical results are impressive, up to 95% editing efficiency in vitro, over 90% mRNA correction in humanized disease models, and durable results lasting 7 to 9 weeks in animals, all at very low doses under 0.1 milligram per kilo. That's remarkable efficiency for an in vivo system. CTX460 and the Alpha-1 Antitrypsin Deficiency market is a big enough market to address, and CTX460 is the first therapy built on the SyNTase platform. It targets Alpha-1 Antitrypsin Deficiency, a serious inherited disorder caused by a single-letter mutation in the SERPINA1 gene. In simple terms, this mutation causes the AAT protein to misfold inside the liver, damaging the organ. And because less functional protein reaches the lungs, patients also develop severe emphysema. There is no cure today, only weekly infusions of AAT protein that treat symptoms, but don't fix the gene itself. So, a true one-time cure could be transformative. CRISPR's goal with CTX460 is to correct the faulty DNA directly in the liver cells, restore normal AAT protein production, stop liver damage, and protect the lungs all at the same time. Now, let us look at what they have achieved so far. At the 2025 European Society of Gene and Cell Therapy, or you call that ESGCT, they met in Seville, and in that presentation, CRISPR presented some compelling data based on animals. Near saturation editing in liver cells at ultra low doses, greater than 90% mRNA correction within 1 week, and 5-fold increase in functional AATD protein, with 99% being the correct M-type protein. No off-target or bystander edits were detected, and I think that's a potential best-in-class profile if these results translate into humans the same way they did in animal models. CRISPR plans to start human clinical trials by mid-2026, and they're currently in the IND enabling stage. And we also should look at the competition for AATD. CRISPR Therapeutics is not alone here. Beam Therapeutics already has its BEAM-302 program. in phase 1/2 trials, making it the first company to show gene correction in humans for AATD. Beam's early human data are absolutely impressive. They achieved therapeutic AAT levels after one dose. They cut the faulty Z protein by up to 78%. No serious adverse events were reported. That's real-world proof that LNP-delivered gene editing can work safely. So CRISPR Therapeutics is now playing catch-up, about a year to a year and a half behind Beam. And so the question then becomes, what does this mean from an investor perspective? Well, Beam Therapeutics clearly has the first mover advantage with actual human efficacy data, but CRISPR Therapeutics could still win if CTX460 demonstrate clear superiority in editing efficiency, protein expression levels, and durability or safety. SyNTase's design with no bystander edits and near saturation efficiency gives it strong differentiational potential in my opinion. And even if Beam reaches the market first, the AATD market is large enough, roughly 250,000 patients worldwide, for multiple winners. Now, for CRISPR Therapeutics, success here would also validate the SyNTase platform for other diseases, positioning it as a foundation for future in-vivo programs. And in closing thoughts, I would sum up everything by saying that SyNTase could be CRISPR Therapeutics' biggest technological leap since Cas9 itself. CTX460 looks promising on every technical front, but it must prove itself in humans to challenge Beam's head start. The race is on to define the first curative treatment for AATD, and whichever company gets there first could set the standard for in vivo genetic medicine. And from a patient perspective, this is a welcome change, and many AATD patients are watching this space to see if they can get a best-in-class treatment and if they can get a choice of multiple approaches. Now, friends, I would also like to give you some more information over and above this. First of all, I want you to understand that I'm a retail investor like yourself. My objective is to share my thoughts and my research out here as a retail investor so that you can get the benefit of all the work that I have put in. And in return, what I want to do is I want you to join ShareTrek, become a subscriber here, and be part of our community, be active in the comment section. What I want to do is create a community of retail investors who can crowdsource, trade ideas, and market intelligence, and collectively we can become much better in the market. And that's what I'm aiming to do. So please take this video in that spirit. I'm not a financial advisor. This is not financial advice. And neither am I a scientist or a biologist. I just put a lot of efforts to understand the companies that I invest in. And I invest in a lot of genomics and AI companies. So I do a deep dive. I use all possible resources that are at hand and try to produce original research out here to the best of my ability. And in case you are an expert in these fields and if you find any mistakes or deficiencies out here, please feel free to put it in the comment section and we all get much better for it. Well, friends, thanks for watching this video. If you haven't subscribed yet, please consider subscribing today and help grow this channel. And while you're at it, why don't you press a like out there, and the YouTube algorithm will really thank you for it and show this video to others like you who might be really interested in it. Well, with that, I would like to end this video here. I hope you guys are having a happy and great weekend. I'll catch up with you again in the next one. And to all the Canadian viewers, happy Thanksgiving. Bye for now.